Hi, I'm Katy Stubbs and I'm going to talk to you today about axonal degeneration, so how axons break down and die. So axons are the long projections of neuronal cells and they carry electrical impulses. So they're like the wires in our brain. And they all join up and form networks like you can see behind me. And these networks are really important. And you can imagine if one of the axons in that network became damaged, broke down and died, that would be quite catastrophic for that network. And that's exactly what I'm interested in. And the problem with axons is they're actually very prone to damage because they are so long and thin. And this can be damage physically, or this can be damage occurring in a disease like Alzheimer's. Now, you may have heard of Alzheimer's disease, and that's because it's the leading cause of dementia around the world. But the problem we have with Alzheimer's is we have no treatments. We do not fully understand why the cells in the brain are becoming damaged and dying in the disease, and so we cannot offer treatments. However, what scientists do know from looking in the brains of people with Alzheimer's disease is the changes to the axons in their brains look very similar to the changes occurring in axons that have been physically injured. So the axons get these really big swellings all the way along their length, and in the axon itself, structural components have broken down. And then ultimately, the whole axon fragments into little bits which are cleared away, and so it's lost. And because this process looks very similar in both injury and in disease, scientists have thought, well, could it be controlled by the same process? Could one mechanism be doing both these things in these different situations? And this is an interesting idea because we actually know a lot more about what happens to an axon when it's been injured because it's been a lot easier to study in the lab. And so the hope is by taking lessons from injury and the axonal degeneration there, we could apply them in disease and therefore be able to find a treatment for Alzheimer's and other diseases. So by studying axons and axonal degeneration in the lab and models of injury, scientists have been able to find mutations which can delay this process, can prevent an axon from breaking down and being cleared away even though it's been injured. And so what my project has done is taken one of these mutations and put it into a model of Alzheimer's disease to see whether it could do the same there, whether it could delay or prevent axonal degeneration, and whether that would provide any benefit to the animal in terms of disease-associated behaviours that we measure. However, this was not the case. Despite putting this mutation in there, it did nothing. The axons degenerated just the same, and the animal saw no improvement. And this is quite an interesting thing to consider, because this suggests that there are actually different mechanisms controlling these two processes, and that we can't simply assume that because the two processes look very similar, that the same thing is triggering them. And therefore, that's why we need to focus more time and energy on looking at disease itself and not just relying on injury data. Thank you very much.